Good morning. Welcome to the webinar, Rethinking Food Safety, Mitigating Risk Through Food Packaging. This event, brought to you by Food Safety Magazine, is sponsored by Aptar. I'm your moderator, Barbara Van Renterhen, Editorial Director of the magazine. Thanks for joining us today. Our presenters are Dr. Jennifer McIntyre, Vice President of Food Safety and Technology at United Fresh Produce Association, and Dr. Angela Morgan, Director of Business Development and Food Safety Solutions at Aptar. Don't forget to submit your questions throughout the presentation. Later in the program, our speakers will address as many of your questions as possible. Today's event is being recorded and will be archived on our website, foodsafetymagazine.com. And now I'm excited to turn over today's presentation to Jennifer and Angela.
Hi, good morning, everyone. Thanks, Barbara. This is Jennifer McIntyre with United Fresh, and thanks, everyone, for joining us, even though things are pretty crazy right now. I think a lot of us in the food safety space have been focusing our attention on coronavirus and the pandemic, but even in a pandemic, we need to eat as a society, and we need to make sure that we, on the food production side, are producing food safely. So we know that coronavirus is not a foodborne illness. I know many of us have been busy trying to communicate to consumers not to use soap or bleach to wash their produce at home. Other things have changed in the food safety landscape. So audits, many of the third-party audits, as well as regulatory inspections are on pause. And we see food companies even changing their operations a bit, having to reduce crew sizes for the sake of social distancing, slowing down line speeds, some even pivoting their businesses, changing formats to accommodate the increased demand, especially in the home delivery side, and the decreased demand on the food service side. Our public health system is clearly stressed, and I am of the opinion that now more than ever, it would be a terrible, terrible time to have an outbreak. So we really need to make sure that we're putting our best foot forward and doing all we can for the sake of food safety. At United Fresh, we represent the fresh produce sector, from grower, shipper, packer, up through fresh cut processor, out through distribution, retail, and food service. And the fresh produce sector has, over the past several years, gotten a lot of attention when it comes to foodborne outbreaks. So I'm sure that many of you have seen the headlines, seen signage within your grocery stores for various advisories, not just for romaine, but other products as well in the fresh produce space. And this pie chart here from CDC shows that whether it's root underground vegetables, sprouts, seeded vegetables, fruits, vegetable row crops. There are many fresh produce items that have been associated with foodborne illness. Why is that? I think one of the, at its most basic level, one of the challenges that we have in the fresh produce side is that there isn't a kill step. So we've got various types of products going through various types of handling, production practices, but nothing, at least so far, that would really be considered that true kill step, that thermal process that can just destroy virtually all microorganisms. We don't have that benefit. So what is used quite effectively, I think, within the fresh produce sector is a multi-hurdle approach, recognizing that at each point, there are things that can be done or things that can go wrong that impact the safety of the product. So at the overarching level, training around food safety, good employee hygiene and sanitation of a facility, all the way through to prevent contamination, and also if contamination occurs, to limit growth. So at the growing side, we've got the input, water, control of animals. Through packing and processing, some operations have wash water. We think about the potential for contamination with listeria from the environment. Even through distribution, temperature control, while contamination, especially pathogen contamination, should not be there in the first place. If it is, temperature can be effective in slowing down the growth. And then even at the consumer stage, and consumer handling. There are things that consumers can do to increase risk or to decrease risk. So there's a quick poll that's up here. I would invite you to submit your comments. How concerned or prepared is your company about potential food safety outbreaks? So are you extremely concerned and extremely prepared somewhat, a little bit, or not really concerned at all? So if you want to take just a couple of seconds, we'll wait here as the numbers are coming in to see the poll results. So we'll give you maybe another 15, 20 seconds or so, and I see the results starting to come in here. Numbers are still going up here, so you've got a few more seconds. All right, we'll close it out here in just another few seconds. Oh, and I guess that prompted some people to get their votes in. All right, well, let's take a look. So it sounds like within this audience, there is a high degree of concern and preparedness as a result around food safety. Not surprising, because it's probably a pretty food safety oriented audience. But there are some that are a little bit on the lower end, but it sounds like many of you are concerned and are looking to remain prepared for a food safety related issue. So as I mentioned before, within fresh produce specifically, there isn't that terminal kill step. Outbreaks, despite our best efforts, are continuing. Consumers and customers are demanding new solutions to prevent foodborne illness. I'll be so bold as to say I'm not sure that the numbers of foodborne illnesses associated with fresh produce have truly increased. I think the visibility, our recognition of it, has increased, and it's become, part of everyday conversation when these outbreaks occur. So we see consumer demands changing, we see brands being affected by these illnesses, and customers asking their suppliers, "What more are you doing? What more can be done?" So, as Angela will talk about a solution.
Protecting the food that feeds our families begins miles away from our dinner tables. From farms and processing plants to restaurants and supermarkets, there are many steps in the supply chain where food can become contaminated by pathogens. But what if there was a way to control these pathogens at the packaging stage, before the food even gets to the consumer? Thanks to Aptar, now there is. Introducing InvisiShield, a revolutionary food protection technology that invisibly helps shield consumers from potential foodborne contamination and illnesses, and aims to reduce food recalls and food waste. This patented platform technology features an antimicrobial delivery system as a final intervention step to any package after it is sealed. Aptar's three-phase active polymer platform technology is at the heart of InvisiShield. It works within the closed package by reducing pathogenic microorganisms like bacteria, fungi, and viruses in the environment surrounding the food. In third-party validated research on tomatoes, InvisiShield technology eliminated 99.9% of pathogens in a range of contaminants, while sustaining the quality, taste, and texture of your food. At Aptar, protecting consumer health is in our DNA. And with InvisiShield technology, your brands, your customers, and your products will be protected like never before. Learn more at aptarfoodprotection.com.
So before we go into more information on the technology, I would like to do another poll just to understand how much background people have on the technology. How familiar are you with VisiShield technology? This is the first time I've heard of it. I saw an announcement about it. I've had discussions with Aptar about it. I'll give a few seconds for everyone to answer the question. You can see the numbers coming in here. Looks like almost everyone's voted. Okay, we'll go ahead and review the results. It appears like most of you, this is the first time that you're being introduced to the technology. So thank you for joining, and I will jump into explaining how the technology works. The video did a nice job of introducing the technology as well as the three-phase active polymer behind the technology. This controlled delivery system has been used in the pharmaceutical space for the last 20 years. And it's been used in a variety of products such as diabetes glucose test strips, where we currently hold 99.9% of the market share. As you can imagine, it's very important for this technology to have excellent Six Sigma processes in order to keep consumers safe. Now it's time that this technology be brought to the food and beverage sector to help protect our food products. How does it work? Well, the three-phase active polymer has three parts. It has the base polymer or the base polyolefin, a channeling agent, and an active component. Once inside a sealed tray, the moisture will build up from the product's water activity and trigger that channeling agent to release the active component, in this case, chlorine dioxide. You can see an image of how the three phases occur inside the polymer in the scanning electron microscope here on the left. This is the formulation that we're going to speak about today for VisiShield. However, there are opportunities to modify the active component and the channeling agent to fit your product's needs. So what else can this technology do? Today we're talking about VisiShield and pathogen reduction that you see here in the left in red. However, there are numerous options. The technology can either scavenge or release volatile compounds. So it's been commercialized for use in moisture control or scavenging CO2 or even removing odors. On the right-hand side, you'll see how this technology can be integrated. Today it's being used, it's being extruded, but it could be injection molded, thermoformed. If you were going to use a bag format, it could occur in a hot melt in the seal. So there's a variety of commercial applications. As I continue through this presentation today, please keep an open mind that this can be tailored to your product's needs, your problems, a specific food type, and it can be integrated into different types of packaging. VisiShield, what is antimicrobial? Well, I already mentioned it's chlorine dioxide, which here in the U.S. is approved and has been used in a variety of different ways. So it's been used to disinfect drinking water since the 1920s. You can also see the variety of other things on the slide that it's used for already in the industry. However, chlorine dioxide is not regulatory approved in all regions, for example, Europe. So our R&D team is working to develop other proprietary blends of antimicrobials to be used as a different active other than chlorine dioxide in this three-phase polymer technology. How is InvisiShield different? You may be thinking, well, couldn't I just gas flush chlorine dioxide into the headspace of a package and it will give me the same result? Well, the truth is that this R&D team's been working numerous years in order to get the perfect dose of chlorine dioxide in the headspace of the pack. And what makes this technology different is the control of the kinetics, illustrated by the tortoise and the hare. So we're able to tailor whether it's a fast release or a slow release based on the product's needs. And it's really a balance between finding the efficacy in pathogen reduction and maintaining the product quality or organoleptics, that flavor, taste, aroma of the product. I've given you a lot of information, but I want to make sure that we touch on what's really, really important for deployment. So first, I want to introduce that one thing that makes this very unique is that this technology is packaging agnostic and machinery agnostic, meaning it does not need to be sold in combination with any specific package or equipment. We can tailor it to your needs so that you can use your existing equipment and existing package. The only requirements is that it's happening inside a sealed environment and that there is a headspace in the pack. So you see here the tray lid combination is what we've commercialized. However, it could be used in a bag format. However, it could not be used in a vacuum package because it does not have a headspace. I've already mentioned that the current channeling agent is triggered by moisture. Therefore, it works best when the product has a high water activity. However, if you wanted to use it on, say, a lower water activity product, such as a tortilla, you could simply have a dampening roller in line that would wet the technology prior to it going inside the package in order to activate it. And the final thing is time. And this will make more sense as we go through the presentation, but we're recommending 12 to 18 hours for the technology to work before it gets to the general consumer. We want to ensure that the product has completely finished acting so that you have the safest product possible. So now let's talk about efficacy, or the actual pathogen reduction from the technology. You see here some of the three most common pathogens: Listeria monocytogenes, Escherichia coli, and Salmonella. This was done by numerous third-party labs, and what you're seeing here is the average of those results. So this is a low inoculation, and we also conducted high inoculation testing to ensure that we could tell how it was behaving at different inoculation levels. Of course, 9 logs is unrealistic, and hopefully you would never see that in a manufacturing facility. One of our customers asked, okay, this is fantastic. It's working for gram-negative and gram-positive bacteria. How does it work on viruses? So this led us down a path of partnership with NC State University and Dr. Jacobs' lab. She conducted a challenge study with human norovirus and found that we were able to achieve a greater than 5 log reduction using InvisiShield technology. InvisiShield is really a broad spectrum product, and we're able to achieve up to a 3 log reduction on gram-negative and gram-positive pathogens, as well as we've tested Geotrichum, which is a fungus, and we can achieve up to a 5 log reduction in viruses. We've tested human norovirus, feline calicivirus, and hepatitis A. I want to stop and spend a little bit of time talking about the way that these were tested, because I think that it really shows the breadth of the technology. So when we first started out, we were working with diced tomatoes in a 5-pound tray. So a lot of surface area and a very dense product. The entire tray was inoculated, but it was important that in our sampling, we were taking from the the top, middle, and bottom of the tray to ensure that the technology was getting every single tomato in that tray. And what you saw on the previous slides was actually the average of the sampling location. Another example is when we tested sliced tomatoes. So sliced tomatoes are stacked up in the tray, and it's very difficult to get to the interior part of the tomato. But we wanted to ensure that InvisiShield technology was reaching that space. So there was internal needle inoculation done, and we were able to show that the InvisiShield technology was reaching the interior part of the tomato. So Jennifer spoke a lot about the hurdle technology and why it's important for the fresh cut industry. This technology was originally developed for the fresh cut industry as there is no true kill step, and we're relying on multiple hurdles in order to have this product possible. So we continue that continue using good manufacturing processes and interventions both in the field and in the processing plant. However, we're now offering a final intervention step before it gets to the consumer, happening inside a sealed container, inside that package, which is preventing it from being cross-contaminated before it gets to the consumer. This gives your company and your brand confidence that you'll have a safer product. Okay, another polling question. So Polly just paid a $25 million fine for a recent outbreak. Is your company equipped to handle a fine such as that? Yes? No? Not sure. See how many people are still with us. Question, answers are starting to come in. A few more seconds here. I still see some people answering. Okay, last chance. And so it appears most of you really aren't sure. I mean, this is the challenging question, right? And hopefully none of you will ever have to face a fine such as $25 million. But it's interesting to see that some people have actually thought about this. So now we're going to move to the regulatory part of this presentation. In the U.S., InvisiShield is generally recognized as safe, or BRAS, and this allows it to be used in food, and we're specifically targeting the food service industry. However, as this technology continues to grow, we do foresee it being used in a retail setting. Therefore, we have applied for FDA food contact notification to be used as a processing aid. This would mean that the technology would not appear on the label. So the next couple of slides, I'm going to walk through the data that was used for that application. First, you see here a headspace analysis. The study was conducted by a third-party lab, SWRI. And you can see that the chlorine dioxide is slowly releasing over an 18-hour period. After that 18 hours, the chlorine dioxide is no longer present and it breaks down into its respective salts. It breaks down to chlorite, and chloride. Well, it's important to note that at time 0 and after the 18 hours, you do not see a significant difference in the amount of salt present in the sliced tomato. You could Maybe say with the chloride levels that there's a slight upward trend, but it's really insignificant. So what this is telling us is that the treated sliced tomatoes and a non-treated sliced tomato are almost identical, and that the general consumer cannot determine a difference between one tomato versus another from a chemical analysis. Another study that was done is a delayed inoculation study. Here, the package was sealed and the technology was inside, and there were multiple time points at which it was inoculated with Escherichia coli. And you can see up until 12 hours, the chlorine dioxide is releasing and at a high enough concentration that it is reducing the E. coli. However, after 12 hours, the E. coli is able to thrive and grow as if it had never been treated. Therefore, it does not have a preservative or a long-lasting effect, and this technology does not need to appear on the label. It's important to note, however, that this is all happening inside a sealed environment. So in the real world, you should not have cross-contamination, and after treatment, you should not have E. coli present in your food. So now I would like to talk about how InvisiShield can be integrated into your manufacturing facility. The goal is for it to be seamless. So what we've suggested is that InvisiShield has an application unit that works in line, so it doesn't require any additional labor or slow the line speed. In a moment, I'm going to show you a prototype machine that will give you a flavor for how it works. However, we have since partnered with tray sealing companies such as ProSeal that have really taken this and made it very simplistic, very beautiful, and takes out any need for additional labor or concerns about line speed. So I want to introduce how it works before I show the video so that you can follow it. InvisiShield comes as a film, and you'll see it on a blue reel, and then it will go down and come in contact with the lidding film where it will be sealed, and then that film will immediately go over a tray filled with product where it can be sealed onto the tray. In the video, you will see it's not taking place inside a dry box, but you know by now that the channeling agent is triggered by moisture and will begin releasing the active ingredient. So what was recommended to us is that the film be shipped in a foil bag, and then when it's ready for use, it be transferred to a dry box on the top of the machine where it will remain untreated, or it will not be triggered prior to use, and you will get the maximum efficacy out of that film. It's also important to note that we have test strips that you can show that your film is still active. So if there's any question whether or not it's been triggered, you can simply test that on the line. This allows for the technology to work in line and have the machine still be able to be clean and sanitized as it normally would in a facility. We also know that in these plants, the relative humidity is very high, and we want to ensure that the film has maximum efficacy. So now let's watch the video very quickly. You can see the blue reel of InvisiShield film. It's coming down, it's being heat sealed, and then it's immediately going over the tray where it's being sealed. So after this technology was launched on March 18th, I began getting a lot of inquiries about whether or not it worked with specific types of lidding film. I want to make it clear that our R&D team has validated that it works with both barrier and non-barrier film. The lidding film can have a high OTR or it can be perforated, as you would typically see in fresh cut products. This is because the chlorine dioxide is actually very heavy. It will sink to the bottom of the tray. It's the same reason that we were seeing such wonderful efficacy in that diced tomato study all the way at the very bottom, because the chlorine dioxide is able to get to every single space within the tray. Finally, it can be used in combination with modified atmosphere packaging. So this is a rendering for what the tray would actually look like and what we're currently using for commercial application. And you can note that the InvisiShield film is registered in the center of the tray, so it's not compromising the seal. And this product was developed for food service. However, as this technology continues to grow, we do foresee it being used in retail, in which case the InvisiShield technology will need to somehow be integrated with the label to have a better appearance for the general consumer. The last polling question: is your focus on food service industry or retail industry? 1, food service. 2, retail. 3, both food service and retail. I'll give everyone a moment to answer. Okay, it looks like most people have answered. And not surprising, a lot of you are both in the food service and the retail space. I want to share with you some pilot studies. We did multiple pilot studies as we were commercializing this product. And the first one was done with Royal Fresh Cuts here in Atlanta. And they actually deployed InvisiShield in their facility and then sent it through distribution to the end user at Emory University Hospital System. Upon arrival, it was unpacked, and there was a triangle study conducted with 9 trained chefs. Here, in a triangle study, you have 2 samples that are the same and 1 that is different. And the trained panelists were asked to be able to identify the different sample. Out of the 9 trained chefs, only 1 chef correctly guessed or identified the different InvisiShield technology tray. However, they did comment that they preferred the InvisiShield technology. This was really fantastic data. We had previously received data from University of Georgia that said the same thing, but it really gave us confidence that the years of research that have gone into getting the kinetics correct in the delivery system of chlorine dioxide was effective, and therefore these trained chefs could not detect an organoleptic difference. So what proven solutions do we have on the market? Sliced and diced tomatoes, onions, pico de gallo, and peppers. However, we continue to get inquiries, and we're open to other inquiries that are not listed here, but our R&D team is focusing on leafy greens. As Jennifer mentioned, there's a lot of outbreaks, fresh and frozen berries, corn, fresh cut fruit such as pineapple and melons. And the R&D team is also investigating different actives and different channeling agents that can be used to help, with these new products using the 3-phase polymer technology. Before I finish, I would like to explain how this technology was actually pivoted. It was launched March 18th during a pandemic, and we quickly realized that there was a pharmaceutical application that could also be leveraged. So we came up with ActiveShield, 1 person, 1 mask, 1 simple solution. You can simply take the ActiveShield strip, and you can put it in tap water and then place it inside a 1-gallon Ziploc bag with your mask or your N95 respirator leave it there for 2 hours, and it'll be disinfected. The initial data that we saw was getting a greater than 5-log reduction of viruses. However, just this week, we received data where we're now getting a greater than 8-log reduction of viruses on these N95 respirators. If you would like to learn more information, please visit ActiveShield.com. And I will, at this point, turn it back over to Barbara, where we can answer our questions on either ActiveShield or InvisiShield, or Jennifer's here to answer general industry questions as well.
Great. Thanks to both of you for a really interesting presentation. We've had a great number of questions come in, so I want to get right to that. The first one is, you've noticed, I guess, Angela, you mentioned that these chefs didn't really notice any taste difference. Were there any odors that they detected?
That's a fantastic question, and let me explain the design a little bit more for how the triangle study was done. So there were treatment trays and there were control trays, and they looked identical in the packaging format. So the InvisiShield technology was applied to both trays, but the active ingredient was not used in the control trays. So that they, upon receiving detected visual difference in the packaging. So then they opened up the pages and they were asked to not only evaluate for color, but also aroma and flavor. So we have data on all 3 of those, and then we also asked them to then, using those 3 attributes, determine which one was different and if they preferred the different sample or not. So there was no difference in odor, taste, or appearance.
Great. Can this be used for organic produce?
That is a great question, and I will have to honestly get back to you. I don't know the answer to that.
So we can stump the presenters. These are really great questions. So you talked about sliced tomatoes. Does it work for whole intact tomatoes?
Does it work? Yes. Do we have regulatory approval? No, because we've gone through the FDA for GRAS status as well as food contact notification status, and a whole tomato is EPA.
Good to know. This is an interesting one. Do you foresee any studies being done for frozen vegetables?
Absolutely. I mean, we're already investigating frozen fruits, so I don't think it would be a huge difference to investigate for frozen vegetables.
So you had shared that the technology is not required to be shown on the label, but would that be a disadvantage, not being transparent about the package, do you think?
Well, I would leave that up to the brand to decide what they preferred. If they wanted to advertise it, I don't think there's any harm in doing that. But as you can see from the data, once the technology is done acting from a chemical analysis and a safety analysis, there's no difference. So that's why it can be used as a processing aid, just as peracetic acid and others that are used today.
Great. So Jennifer, how do you see this changing really what growers are doing, what produce processors are doing? Do you think this is a game changer for the pathogens that people are dealing with currently?
Well, we always maintain that the focus should be on prevention. That said, we know that doesn't always work, and so any additional hurdles that can be shown to be effective for that particular matrix, I think it's up to each company to then evaluate if if it's suitable for their product and for their application. And as Angela said, on the grower side, when we're dealing with raw agricultural commodities, that is kind of a different framework. And there are just different opportunities and different limitations associated with raw agricultural commodities versus the processed side later in the supply chain. So from my standpoint, and within United Fresh, we're always interested in seeing what the newer technologies are, what the different products are, what the different applications are, and of course, what the science says in terms of the effectiveness. So to me, the big game changer would be like a total kill step. And Angela said this is not meant to be a kill step. It's meant to be an additional hurdle. So I think that that's great. And that's the space where fresh cut processors in particular, it sounds like this is an area that could be explored by them in the short term.
Great. So we're talking about produce, but we're getting some questions about applicability to other products, such as fresh or processed meat. So do you want to take that one, Angela?
Sure. So this is an R&D area that we haven't begun working in yet. However, we are looking for partners. We want to understand the pain points in the seafood and meat industry, specifically what pathogens you would be interested in targeting. And then we could have the R&D team investigate how the technology would perform. We could test the formula that we discussed today, or we could look at other actives, to see how they would perform as well.
That's great. So this is an interesting science question. So how about potential pathogens on the surface area, so maybe the stacked area of the food products that are not directly exposed to the chlorine dioxide? What is the penetration rate of the gas through the food products?
Yeah, so that goes back to the way that we did our testing. So the internal inoculation that was done with the needle inoculation, ensuring that it was getting in the inside of the tissues of the products and not just the surface.
All right. So this is an interesting question. This participant uses polypropylene bags, and the ink is a non-food. So what can this person do if they don't know the ink migration rate? Would that be affected?
I assume that the bag is printed on the outside and that this technology would be taking place on the inside of the bag where the ink would not be present. But I would have to talk to that individual to understand the problem in more detail.
So I've gotten a couple questions about possible cancer risk from exposure to chlorine dioxide. I'm sure that you've looked at the health effects of this agent, but if you say a few words about that.
Sure. So in order for us to get the GRAS status as well as apply for the food contact notification, we've been working with a third-party company called Toxel that specifically evaluates the toxicity of any antimicrobial that we're evaluating. And they're the ones that actually hired SWRI to do the chemical analysis that you saw just a brief synopsis of in this presentation. But there is entire toxicology reports, and they've been evaluated by numerous experts to ensure that the product is safe and that the government would give us that GRAS status.
Great. So when you're adding this to the package, does that affect how someone would be able to recycle their bag or their container?
So that's a fantastic question that we've been evaluating in great detail. And it's one of the reasons that we've decided to make this product packaging agnostic, meaning it couldn't be used with any package. So, it could be used on a biodegradable tray, for example. And the only thing that right now, until we are able to find a way to have the actual strip recycled, The best thing that we can offer is that it has an adhesive that can be removed, and therefore you can continue to recycle the lidding film and the tray or the bag or whatever you're using, but simply that one piece would not be recycled. But our R&D team is working on understanding how the technology itself could be recycled.
Great. Is there any limitation to how quickly food could be consumed once exposed to the gas?
It has a normal shelf life, and we validated it over a variety of different shelf lives. So for diced and sliced tomatoes, for example, we've evaluated the technology's efficacy over a 14-day typical shelf life for that product. It is working, at least in that formulation, very rapidly in the first 12 to 18 hours. And then the shelf life is maintained as usual. So, the consumer could consume the product anytime after it gets to its endpoint of use. This technology is really being deployed during distribution.
Great. We have a participant who is in South Africa and wonders if the technology is available globally.
Our focus has been on the U.S., but I'm constantly having questions from a variety of different countries. This is the first request from South Africa that I'm aware of. But I do believe that we could look into the regulatory requirements in South Africa, and if it meets the minimum requirements, then yes, this technology could be deployed there.
Great. So I'm going to read, this is a rather long question, but it refers back to the studies that you talked about. So the person writes, it is critical that the inoculum be allowed to acclimate to the produce, which takes no less than 24 hours up to 48 hours, and that even water is effective at dropping the inoculum numbers several log cycles during that period. But that looks like when you're reporting the bulk of the drop in numbers. So can you address that concern?
Yeah, so first I want to say that I'm in sales and our R&D team can answer all of these questions. But I do have an understanding that these were done by third-party test labs where the bacteria was appropriately attached to the food matrices. It was allowed a drying time, and we always had controls. So if there was an inherent drop that would just naturally occur, it would have been seen in the control tray. And you can see that the log difference that we're reporting is based off of those controls at the same time point.
Great. This participant mentions that their company has a repacking facility, so they don't work with ready-to-eat produce, but they do handle the fresh produce in order to repack it, and wonders if they can apply this technology to their process and how they would do that.
I would need to understand more about the process, but I have had some conversations with, for example, fresh berry producers where they're actually packed in the field. And one deployment method that was discussed there was there's actually tents that are used that you could deploy the InvisiShield technology inside the tent to treat multiple packages at the same time. I'm not sure if something similar could be applied here, but we could definitely brainstorm to understand how the technology could be modified for that need.
We've had a couple questions about cost. Are those questions you'd rather take individually with people?
I'm happy to share what's commercialized, right? So the Emory University Hospital was paying $1.07. Obviously, they had a really great value proposition because they're able to now service immunocompromised, like cancer patients and other people with fresh products that are very nutritious. So there was a fantastic value proposition in that case. And for the Active Shield technology, it's been commercialized at $1.13.
That's great. Wow. Does the acidity of the product affect the process? I mean, tomatoes are fairly acidic, so.
Yeah, it's a great question. We've evaluated that. Toxel helped us out greatly with this, and there was a difference in the the reduction that was seen in the formulation needed to be varied from tomatoes to say other products due to that acidity. However, it can be used on non-acidic products as well.
Great. How long is the efficacy, and what is the possible shelf life extension when using this?
So as you can tell, we're not making shelf life claims. We're specifically focused on pathogen mitigation. And we have determined that once you reduce these pathogens, they've been reduced at least 3 to 5 logs, and you shouldn't have that high of contamination to begin with because you've been using all of the other hurdle approaches that Jennifer spoke about. So therefore, we found that the pathogens aren't injured; they're not regrowing over time. So therefore, assuming you don't have any cross-contamination, your product would maintain that safety throughout the shelf life. But we're not making shelf life claims, specifically because we've applied for the food contact notification.
Great.
We want to remain off the label. We're not prescriptive.
Sounds like a good idea. So there are antimicrobials used in fresh-cut produce plants, and so do any of those interfere with your technology, like hypochlorite?
Oh, fantastic question. Yeah, no, I've gotten this question from a variety of fresh cut processors, and we've validated it with their different interventions, such as their chlorine washes or PAA, and there could be a synergistic effect. There has not been any antagonistic, effects observed.
Now, you've mentioned, the toxicology reports. Are those available for review?
I don't know why not. Yeah, contact us. We can definitely provide them.
Great. So I thought this was a good question. Is there any plan to expand this technology to low moisture foods, or even some fruits and vegetables that the quality would be adversely affected with excess moisture?
Well, it doesn't need a lot of moisture, and as I described earlier, we were working with tortillas where the water activity is very, very low. But just like in the active shield, the masks don't have inherent moisture. The only thing that's important is that it triggers the initial release, because once it's started, it will continue acting. So if your product does have low water activity, that's not a problem. You just need to activate the film organically. So, wetting it on a damp roller, or in the case of active shield, dipping it into tap water.
Right. Well, that's what you described, similar technology for the masks, right? You had to add
Correct.
Some water. Do you have any data for spore treatment? Does it affect spores?
Yes. We have not done spore data for on food. So I'll just say that. But we are applying for EPA approval with active shields to be able to clean other surfaces other than just N95 respirators. And so we have lots of literature that shows that it is effective on spores, and we're continuing with that research right now.
Great. Do you sell this in die-cut teardrop or just in bulk rolls?
Right now, just bulk rolls, but we're just launching this technology, and there's a variety of ways that, specifically for retail, we need to be creative and use different dyes to make it more visually appealing.
Good to know. This is a question I wondered about this as well. Some of the produce. Now you mentioned you're working on berries right now, and a lot of time the clamshell containers are vented. So I'm assuming that this technology needs to be completely closed for it to work. Is that correct?
We're in the process of validating that. Like I mentioned before, if the holes are at the top of the clamshell, it's not really an issue because the technology is placed at the top, and the chlorine dioxide is very heavy, so it will sink to the bottom of the tray. However, if the clamshell has holes in the bottom of the tray, then that's where that tenting scenario would work best. But a lot of the berry producers that I've talked to, they're interested in also modifying their packaging format in order to be able to utilize this technology.
Great. This is a question related to the masks that you talked about at the end. Are there any residual gases within the mask after that 2 hours that a wearer would need to be concerned about?
No, so that's actually why the 2-hour period is in place, because the treatment's actually taking place in 30 minutes, and then we're allowing the next hour and a half for the gases to dissipate before you open the bag. And that time point was done through third-party testing where we were able to identify that after 2 hours, people weren't able to detect an aroma. And it went through a fit test where, it was ensuring that the masks are still performing the same, where there's actually like an aroma spray. So there was a whole bunch of work done around ensuring the safety of the mask after it's been treated.
Here's one that I thought was interesting. Is there any effect of light exposure on the levels of chlorine dioxide in the package?
Yeah, I've gotten this question before, and I'm not sure that we've done enough research, but we certainly have retail display cases where a lot of the studies have been done, and it hasn't seemed to have an impact, but I do believe further validation is needed in this area.
Right. And I think our final question, this is a good one. Do you see any bulk applications for this technology? For instance, a supplier packaging tomatoes whole in bulk before shipping to a consumer or processor?
Yeah, so the focus has been on food service, so a lot of the applications are already in bulk trays. But as far as the whole tomato would go, or like I was speaking about with the whole berries in a tent, that would be a bulk application, but that does require EPA approval. So we would need to begin that path.
Great. We do have other questions, and all of your questions for those who submitted them, they'll go to Angela and Jennifer, and they will be able to reply to you directly. So that's all the time we have for questions today. Please join me in thanking Jennifer McIntyre and Angela Morgan, as well as our sponsor, Aptar. If you have any additional questions or comments, please don't hesitate to click the Email Us button on the console. At the end here, our survey will appear on the screen, and we'd appreciate your comments. Please visit foodsafetymagazine.com for an archive of this presentation, as well as information about our upcoming events. We appreciate your time today and hope you have found this webinar to be a valuable experience.